Welcome to Stand Up and Scream. I'm your host, Desiree Rose, and today I'm here with Carlos Gutierrez, who is a chemist, a professor of chemistry, and founder of the MORE program, which is, MORE is?
The Minority Opportunities in Research Programs.
Okay, tell us about yourself. How did you, I know you were an artist, and then you became a chemist. That's quite a leap. How did you do that?
It's actually very, very similar. When I was an undergraduate, I realized that I needed to know some science in support of art. I was a painter, and I thought I needed to know something about my medium. I needed to know why paints were the color that they were, why we use gesso on a canvas, and what the heck was gesso anyway. And so to learn something about the materials of art, I figured that I needed to know some chemistry, and I signed up in
So you took
Chem
a chemistry
1A.
class?
Yeah, at UCLA.
And that was the end of art?
No. I had a love-hate relationship with chemistry. I thought it was wildly exciting and terribly dull at the same time. So I would leave it and then go back, and leave it and go back, and went back and forth between art and science for all the time I was at UCLA.
And so finally, science won out?
Science went out. I found through several people that I met there, faculty members, notably Chris Foote and David Evans, how creative scientists could be, particularly chemists could be, and particularly synthetic organic chemists could be, and it just knocked my socks off. So.
Is that your field,
Yes,
synthetic
yes.
organic? Tell us a little bit about that. I have no clue what that is.
It's that branch of chemistry that is concerned with making molecules, making them elegantly, making them in beautiful ways. And so to me, these people were talking in aesthetic terms. They were talking about economy of steps, simplicity. And so to me, that just resonated, and it made just a whole lot of sense to study that part of chemistry. I'm not that good a chemist that I love all chemistry. I love what I do. I love the type of chemistry that I do. It curls my toes, and so that's why I chose that particular.
So do you make
branch.
some pretty cool compounds?
Oh, we sure do. My students and I, we make compounds that hopefully will be useful as magnetic resonance imaging molecules.
Oh yeah, I know about that.
Yeah, and
Okay.
And my students are using very creative methods to make polyfunctional molecules, where hopefully we'll be able to have domains that will specifically target particular tissue types, and other domains that will work with the solubility of the compounds, and at the same time have some domains that will actually do the imaging.
So what is your recent project? What are you working on?
Well, that's it. That's the most recent one is that we're working on that, and it all revolves around. We have a polyamine, which is a molecule that has many nitrogen functionalities, and being able to selectively address the different nitrogen atoms is hard, but they've come up with a really slick way of doing that. And so to me, that these kids can show their creativity and use their talents, really sculpting at the molecular level, is wildly exciting for me.
And organic, doesn't that imply carbon?
Oh, absolutely.
So
Absolutely.
how does that come into your?
Well, the core of our molecules are built of carbon, but the functionality comes through some nitrogen atoms that we have very deliberately designed into these molecules, and we use them as handles to attach all these groups that will do the targeting and the solubilization of our molecules.
So what is nitrogen good for? Is that
Oh.
fertilizer? I mean, I don't. What do we use nitrogen for?
The properties of nitrogen are wild and varied. All proteins contain nitrogen atoms. It's just a building block that is incorporated into organic molecules, particularly biologically interesting molecules. So we need molecules that will be compatible with biological systems, and so an awful lot of functionality can be had through the judicious use of nitrogen atoms, and that's what we do.
I guess what I'm getting at is like, how can it benefit me or us?
Well,
how can we use it?
Think of what we're really trying to do at a fundamental level is learn how to very specifically target molecules to particular tissues or tissue types. If we could learn how to do that, wouldn't it be great if we could cut down on the dosage of drugs, let's say of a drug or a diagnostic agent, so that this thing would not go all over the human organism, but rather target that particular cancerous tissue, or hopefully even sooner when it's just a cell, or even sooner when it's just starting out and expressing a particular protein that is indicative of cancer.
So you could potentially make chemotherapy a lot less painful and gruesome to go through.
Well, and again, that's a long-term goal, but if we can do it for diagnostic agents where we can image things, should we not be able to do it for therapeutic agents? And there's a whole branch of chemistry now that is looking at molecules that will have both diagnostic and therapeutic properties in the same one. And
so you could find out you have cancer and cure it at the same time.
At the same time.
Awesome.
Is that cool?
Yeah, that's really cool.
So that's a long-term goal that we.
So presently, the chemotherapy at the present time doesn't really target the specific cells. It pretty much
not
goes
with the
where
selectivity that we need. Some of it is quite specific and quite selective, but it's hard to program that into the molecule.
I guess we don't want our hair to fall out, really.
No, see, that would be a side effect.
Right.
What we want is for this very toxic chemotherapeutic to go specifically where we need it to go, and not anywhere else. And so we need to become much more selective, much more creative in how we look at this, so that we can cut down on the amounts of material that we take, so that we can just be much more deliberate about these molecules that we introduce as therapeutics.
God, that would be great if you guys succeed
great.
in your research.
And again, there are many groups across the country that are looking at this from different aspects, and that's what's cool, is that we look at it based on the personalities and interests of the folks in my research group. We look at it in a particular way. Somebody in another laboratory across the country will look at it from their own perspective, and we're going to come up with some great molecules.
So have you had a little success at
well, we're still at the very beginning stages,
genesis
and yeah, but we are learning how to address all of these nitrogens, which is the key part. Is if we're going to be able to put different functional groups, different functionalities, I should say, on molecules deliberately, if we're going to make a Lego set of molecules where we can decide what it is that we need in terms of functionality and snap onto some kind of a common core, these other molecular fragments that will give you that functionality, that's the grammar that we're learning right now. And once we learn the grammar, we'll start putting words together, and then we'll start writing poems.
Oh, that's going to be exciting.
Oh yeah,
huh? That would be great.
It's great.
Now, you also are very active in the MORE program, which stands for
The Minority Opportunities in Research.
Okay, so that's really fantastic. Tell us about how you came to do this.
Well, as a professor of chemistry and as a professor at Cal State LA, 80% of the students at Cal State LA are minority students. Cal State LA is going to look the way, California is going to look the way Cal State LA already looks in 50 years.
Doesn't California look like that already?
No, we're at about 52% minority, which is
we're pretty
already
diverse.
huge.
It's pretty great, huh?
It's great. That's one of the reasons I like living in California.